right, I'm pleased to present our last speaker for this afternoon, Dr. Peter McCourt. He is a professor in cell and system biology from the University of Toronto.
Great, thanks very much. Okay, I'm going to use this mic. I just got to make sure I don't fall off this thing, right? It's kind of scary up here. Okay, I'd like to thank the organizers, particularly Maurice, for inviting me to give a talk. And you know, my parents immigrated from Ireland many years ago, and we immigrated to Saskatchewan, to a little town called Ituna, which is just about 80 miles away from Regina, and spent about 9 years there before we moved on to Edmonton. So it's nice to be back in Saskatoon. And so what I'd like to talk about is what I call trying to understand the drought code. And so maybe unlike many of the talks I've heard so far, which are wonderful examples of, I think, basic research becoming practical, I'm going to talk mostly about basic research. And so can I have that first cartoon there? Turn that on. This is taken from NASA data. This is basically what's going to happen to North America in terms of drought over the next 100 years or so. Okay, and for Canadians there, it was hard to see in the beginning there, but the soil moisture content or in California now is what Saskatchewan's going to be like in 2100. So I don't know if we'll have the California weather, but that's the situation. I feel sorry for Mexico. It doesn't look too good down there at all. But obviously it's a prediction, but as we've heard a lot in this meeting, we really do have to think about the future. So let me see if I can get this right. Is it this? Which one is it? This one, maybe. There we go. Just recently in Missouri, and whenever I go to Missouri, I try to track down the great John Boyer. And John always shows me this slide. 1982 paper from Science that I just found fascinating work. When John was at the USDA, he basically looked at what would consider taking all the data that the USDA was generating and looking at optimal yields versus what we see in the field. And so here's kind of the optimal, here's what they were really seeing. And then, you know, parsing through all that data, he basically came up with where are we losing our yield. And what you can see is you've got obviously disease, insects, weeds, and others. So this would be abiotic stress. And 70% of the yield loss was occurring here. And what was interesting, what John tells me is that this 2% that you see here in weeds, that's what the USDA was mostly funding at that time. And the weed scientists at the USDA actively politicked the Congress to suppress this paper. Okay, so just give you a sense of, you got to be careful. Anyway. You know, as we've heard in this meeting, abiotic stress is a major problem, is the one we have to really think about. If I look at that, I just showed you North America, but obviously this meeting's about developing countries also. I'm just going to quickly take you through what's going on here. Go back on that again. So this is obviously projected soil moistures, again, over 100 years, and you can see that there's going to be major changes going on. So we have a serious problem, which I don't think I need to convince the people in this room about. We have always been interested in drought, again, from a mostly basic perspective. And people always show pictures like this, you know, with a plant growing out in the field. But this isn't the kind of drought that we're going to have much effect on. All right, it's kind of like, you know, years and years of drought, finding a plant, you know, we're not going to be able to grow crops in that situation. So droughts come in many forms. And the droughts that we're really talking about, of course, are episodic droughts. And what that means is they come and go. And if you can just run this cartoon for me, I find this kind of cool. This is satellite data out of NASA also. And it's just showing the soil moisture content in the Nile, kind of the region of the Nile. It's just kind of cool to watch, isn't it? And you can see, you know, you're going up and down all the time. And this is something that I think plant scientists can deal with. Obviously, this is just looking at weather, but you can see that thing, you know, it gets wet, it goes dry, it gets wet. Some years are good, some years are bad, etc. And this is the kind of thing we can have an impact on. Okay, great. So what are our solutions to episodic drought? And we see from this meeting and from obviously reading that it's going to take a multi-pronged approach. We need obviously changes in farming practices, changes in social, political, and of course we need science and in particular breeding, and we've got two kind of ways to go, traditional or genetic engineering. I include in genetic engineering there, I include gene editing, because I'm unconvinced that people will believe that gene editing is not genetic engineering at the end of the day. All right, so we're going to focus on the science. Can crops be bred for drought avoidance? Yes, of course they can. Can we do it logically? I think we've seen today that with molecular technologies, we're getting much more logical in how we develop those crops. And finally, what I'm most interested in is, can we define a genetic code at the basic level? So, what do we know about drought? If we keep it very simple, obviously, if we're going to improve drought avoidance or drought tolerance, we could see how we could affecting root architecture would be useful. Obviously, controlling stomatal pore apertures would be great. A lot of people talk about looking at gene expression because we have tools that are very powerful for doing that. Gene expression, though, is an interesting one to us. This is a piece of data that we generated a number of years ago. What I'm just showing here is this is only about 250 genes, but what we're looking at is different tissues. We're looking at shoots and roots, and then we're looking at different times of osmotic stress or salt stress, cold stress, oxidative stress, heat. And what I really want to show here is this is very complicated. There's differences between tissues. Some of the abiotic stresses kind of cause similar patterns, but others don't. And obviously at different times you get different results. And so the issue with just using transcriptomics or gene expression is it's very interesting data, but this type of analysis really doesn't have any functionality to it. Okay, so it's hard to see what's going on in these regions. That's where, of course, we need systems biology and people like that to tease out, are there patterns in there? So, of course, the other approach is mutant analysis, whether it be for genetic screens by traditional mutagenesis or through breeding. If I can just run this, this is a list of genes that have already been tried in rice. Okay, it's from a review by Shinozaki. So, there's lots of genes out there. Every one of these genes was thought to be important in drought. Okay, and maybe some of them are, some of them aren't. So there's lots of genes. We've got that already. But the problem is that, and this paper really put an earthquake into our lab. This was a paper out of Dirk Inz's lab, he published in Nature Biotechnology. And what Dirk did is, for the Arabidopsis people, he took 25 Arabidopsis genes that had been touted as being drought tolerant, improving drought tolerance in Arabidopsis. And he did very, very careful analysis. And instead of doing it the way most Arabidopsis people do, where they just stop watering their plants, what Dirk's group did was they did a mild drought stress, something more physiologically relevant. And so we're looking at these 25 lines. And what he found was that none of them were drought tolerant. Okay, so there was no correlation between all these plant cell papers on showing a green Arabidopsis plant in a pot, and actually when you actually put it in not a field situation, but more of a normal situation, you don't find any correlation at all. Okay, so it's worried us, I'll tell you that, right? So we really have this issue. We can generate mutants in Arabidopsis, or we can do transcriptomic studies, but how do we find that signal in all that noise that we're dealing with? So one thing I think that is important is that we do need to start making a code. We need to start putting these things together into pathways, et cetera. And so this is again taken from a Shinozaki review showing, kind of in a general sense what's going on. It's more like what's looking more like a subway map now that kind of gives, that we can hopefully generate hypotheses. I think we just saw from the previous talk that Leon was showing is that if you start out with aluminum toxicity, but we're now starting to get to some kind of map of what could be going on here. And so the area that we're mostly interested in has been abscisic acid, and this is the abscisic acid signaling pathway that we've been talking about that's been worked on by a lot of people. One of my former graduate students, Sean Cutler, was involved in that seminal work at cloning the ABA receptor. So why, let me go back there, why work on, so if we quickly talk about ABA signaling, obviously here's the molecule, it binds this receptor, and then the receptor inhibits these PP2Cs, and that allows these SNRK2 kinases to activate transcription factors, and you get expression. So we have a nice, simple model that we can test. And why would we work on ABA? Well, physiologists have shown us for years and years that ABA can improve the drought tolerance of a plant. It's just a tomato plant, and obviously they've drought stressed it in the absence of ABA or misting it with ABA, and you do get improvement. So we have a pathway and we have a phenotype. So it seems to me like it's a useful thing to go after. So again, just demonstrating the ABA pathway that Sean and many other people have been involved with. ABA is transported through, binds its receptors. It's quite complicated. There's a lot of redundancy at the receptor level. These receptors normally will inhibit these PP2Cs, which are inhibiting these SNRK2 kinases, which eventually, once ABA binds, the SNRK2s are released and you get activation of transcription. What I think maybe is very exciting at some level is that when we over or underexpress various components of the ABA pathway, we do get improved drought tolerance. If we overexpress the receptors or underexpress the PP2Cs, overexpress the kinases, and even some of the transcription factors, we get logical outcomes. And that's not just in Arabidopsis, that's been found in other plants where that's been tested. But I think we all know that the world is going to be much more complicated than just core signaling pathways, and that what we've been trying to do lately is turn these core pathways that people like Sean have generated into ABA networks to give us more genes to think about that might be useful for breeders. So how do we go about doing that? Now, when people talk about networks, they can get so complicated. You get these big hairballs of networks that you really, when you look at them, they're very exciting, but then you think, I don't understand what the hell is going on. So we took a very focused approach that was based on a paper by Mark Vidal and George Church in yeast, in which they showed that transcribed genes that are co-transcribed, more often those protein products interact with each other than a random data set. Now, when you hear that, you say, of course, that makes sense, right? That when you turn on, so you give the organism some kind of event, the genes that come on, yes, they make proteins that interact. That's not surprising. But of course, before they actually did the experiment, I had never really thought about that. So what we did is we did the following. We said, okay, we're only going to collect genes that are transcriptionally regulated by ABA, up or down. So we're not going to look at all these other genes that might confuse things. And then you might expect genes that are transcriptionally regulated by ABA, there's going to be a chance that a number of those will actually interact at the protein-protein level, which is what's required for a signaling pathway. And once we define that signaling network, we can start to make gain and loss of functions of it and see whether we get similar phenotypes, ABA-related phenotypes. And that was kind of the logic, these 3 steps. So we did that, and this was all work done by I'm sorry for John here. I'm not going to talk about strigolactones at all, or Striga. I'm going to talk about ABA. This was all done by a research associate in my lab, Shelley Lumba, who's continuing on with the ABA work in a big way. So what we want to do here is we want to turn this core pathway into a network. So Shelley did she cloned 250 genes and did 250 by 250 all-by-all interactome, which is about 65,000 potential interactions. And what she found was about 520 interactions over 140 genes once you do all the bioinformatics and all the systems biology that's involved. And so what do those networks look like? So we have a core. It goes to something like this in terms of just the core alone. So what you can see is we have a lot of things that are transcription factors. Where you see red, that means red means that when we did genetics on those, we got decreased ABA sensitivity, and where we did genetics on the blue ones, that's where we got increased ABA sensitivity. And so we're starting to now look much more like a network rather than actually just a simple core. Now, these are just genes that touch the core. Okay, but if we look at the network itself, it looks more like this. And it's highly enriched for, in yellow, are transcription factors or things that are thought to be in the nucleus. So that's our kind of mini interactome, if we want to call it, or a mesoscale interactome of about 140 genes, including the core, and how those proteins touch one another. So how can we use that now to attack drought? So now that we have a network, we can look at subnetworks. Here's a subnetwork. that we were very interested in. A lot of transcription factors, or a lot of nuclear localized proteins in there. Okay, what we did is we took all these and we fused them to a DEX-inducible promoter and put them back into Arabidopsis. And then we looked at their stomatal conductance. And so many of them, so we're looking at, this would be, we're activating the gene with green, or we're doing nothing. Many of them, let's say they don't have any effect. But this one here is really intrigued us, so that would be this guy down here. So you can see that when we activate the gene, the plant loses water out of the stomates a lot slower than if we don't activate the gene. Here's one that didn't change. Here's one that went the opposite direction. Okay, so you can go on and do this, and because we're only working with 140 genes, we can actually do this kind of genetics. If someone hands you 1,000 genes, you just don't know where to start. So what we did then is we took that one, and instead of using a DEX-inducible promoter, we actually used a Drought-inducible promoter. So now we're taking it turned out that little node there is an AP2 transcription factor. We're now expressing under a promoter that's induced by Drought. Here's wild type. And here is our Drought-inducible plants. What we found interesting about this was the following. So if we took that gene and put it under 35S, we got no drought tolerance. All right? So I would warn people out there that I think regulator promoters, and we've heard this from other people, regulator promoters are going to be a very important component of manipulation of plants. That if you just constitutively overexpress the same gene, you don't get any effect at all. But if you put it under drought promoter, we did get an effect. So I'd like to encourage people to think more about their experiments than just using 35S. Okay, back to Durkin's little earthquake paper. So it made us think, now unfortunately, I work in Canada, so I have a small little NSERC grant. So I can't do this kind of fantastic stuff they do there in Belgium. So we thought about, well, how could we do these experiments on Canadian money? Okay, Canadian Tire money, I guess. All right, so Shelley came up with the following. She said, you know what we're going to do? We're still doing Arabidopsis, but we're going to just do it in a conviron like everybody else. We're going to take, let's say, I'm going to show this example here. 6 trays, 18 pots in each tray. We're going to decrease the water. We're going to use constant volumes of water. We're going to move the pots around just by undergraduates, etc. And so what we're looking at here is so this would be an example of where we water the plants with exactly the same amount of water every 2 days, and this is obviously the time. So going from 19 to 46 days, for example. This would be a situation where we only watered every 4 days, every 6 days, every 8, every 10, every 12. And so you can start to see that obviously when you're only watering these plants once every 12 days, you can see that they're quite sick out here versus the controls up here. But what intrigued us about this result was that here's an example of if we only water every 6 days, you can get quite a reduction in seed weight, relatively quickly. But if you look at what the plants look like, they don't look that bad. All right, so they're not these yellow things, but we are getting a dramatic decrease at day 6. We're getting a 50% drop in seed yield. All right, and I think this goes back to what Dirk's saying is that Arabidopsis people are screening for green plants in a yellow background, but actually, even with their staying green, the yields aren't that great. And so maybe that's one of the reasons why we don't get this translation of these results that we see in journals like Plant Cell into the field. So just another example. So what Shelley did was she took we collected a collection of knockouts in our network. So now we're looking at seed yield. I think that's right. Sorry, that was the white lines were actually rates of water loss, and the yellow lines are seed yields. And so you can see, actually, if I represent that another way, there was no correlation we found between whether the plant stayed green and whether its seed yield was any good, except for that guy up there, which is the one I've been showing you. All right. So these were all loss of function mutations, in which it's just like throwing darts. But obviously, when we did drought-induce overexpress this transcription factor, we did get the kind of result we expected. So I think there's some lessons in there somewhere. I felt I better show some translational work. So we took that AP2 transcription factor, put it in soybean. This is very early work. I mean, we don't have enough. We don't even have close to enough population studies. But what we found was when we did it, when we made the same, redid the experiments with transgenic soybean, we do see something going on, and I'm not as happy as what I'd like to see. But basically, what we're seeing is obviously this is our one of our soybean plants, wild type. Here we've drought stressed it. I didn't know. It's the first time I ever worked with soybean. I didn't know it was so sensitive to drought compared to Arabidopsis even. But here, obviously, the wild type's getting pretty droughted, a little bit better. And then, obviously, when we rewater, you can see that the transgenic is doing better than the wild type, which is now starting to come back. So we're buoyed by that. We're thinking, okay, so we did get a situation where the Arabidopsis work has, at least in the greenhouse, translated into soybeans. So maybe by being a little, I'll be honest, as one of these callous Arabidopsis people, maybe Arabidopsis people need to do their experiments with a little bit more physiology and talk to the people like John Boyer, the plant physiologists, about how to do a good drought experiment before they try to make the cover of Plant Cell with their drought-tolerant Arabidopsis plant. So that's certainly the lesson that I've learned. So I think that's about all I've got to say. Here's the lab. Like I say, all the work was done by, there's Shelley there. And unfortunately or fortunately, we're moving on into strigolactones now, and I'll have to give that talk another time. So thank you very much.